I'm very happy to present here today. So our story begins a few years ago when Barbra decided to clone her dead dog. Yeah, she loved that dog, and in order to do that, she needed to take the cells, and then the scientists extracted the DNA from the cells of the dead dog and put it inside a fresh egg. What we are going to show you today is a collaborative project that will allow us to take cells from skins in general, and to produce all the cells required to produce an artificial embryo, and all the cells supporting this embryo. To do that, we need a collaborative environment envisioned by Yossi Buganim, Dr. Yossi Buganim.
Hi, so how does life begins? So when a sperm fertilizes an egg, it creates a unique cell type that's called zygote. This zygote holds the capability to produce all the embryonic and the extra-embryonic tissues. Upon division, this capability is gradually lost until a clear segregation is seen in the early embryo. We can see here 3 cell types. The green cells, these cells are the stem cell of the placenta. We have the red cells. These cells are the stem cell of the fetus. And we have the blue cells that can create the yolk sac and umbilical cord. Today, we know to take these different compartments and culture it in the dish. We can take again the red cells and put it in the plate, and we have fetal stem cell and so on for the placental stem cell and the yolk sac stem cells. A year ago, a really amazing groundbreaking discovery has been made by 2 British scientists. They showed us that it is possible to take placental stem cell and fetal stem cells, combine them together in the culture, in the plate, and these cells knew by themselves how to reorganize themselves into an artificial embryo, embryo-like structure. Here in the left, you can see the synthetic embryos, and in the right, you can see an embryo, a real embryo. However, there are few limitations for this technique. One limitation is ethical issues. In order to build this synthetic embryo, you first need to kill an embryo to isolate its stem cells. Another issue, another limitation, that these scientists used only 2 stem cell types. They are still missing the cell type that generates the yolk sac. And maybe most importantly, if you are going to use this technology to model diseases like placental insufficiency, once you know that you have a problem in the placenta, you don't have any more stem cells. So these stem cells are not available for us anymore. So we still have the same problem of how can we take this stem cell from a developing placenta. And this was really our goal in this research. And our discovery was made firstly by an amazing groundbreaking discovery made by 2 Japanese scientists, Takahashi and Yamanaka, who taught us in 2006 that you can take a skin cells, introduce into these skin cells 4 genes, and after 3 weeks, you can get fetal stem cells. These fetal stem cells look like and behave like exactly like embryo-derived fetal stem cell, and you can also generate an entire mouse from these cells. However, these cells cannot produce placenta and umbilical cord. So as I told you, in 2006, they taught us how to take skin cells and produce fetal stem cells. When I opened my own lab in 2014, my first goal was to recapitulate this process, but this time to take the skin cells and to try to develop placental stem cells. And indeed, this is what we have done. So we know now how to take skin cells, convert them into fetal stem cell and to placental stem cell, but we are still missing the third part. And this is actually the goal of this study that we present today. We aim to take skin cells and to find one combination of genes that can produce all the stem cell types at once. In order to do that, really, this is really challenging question. In order to do that, we had to build a sophisticated reported system, a system that will allow us to distinguish between the three stem cell types. For that, we engineered the skin cells to hold a specific flag. We use several flags, and I will give you two examples here. So, for example, if these skin cells will turn on three flags, for example, the red, the yellow, and blue, we know that we succeeded to convert these skin cells into placental stem cell. In contrast, if another skin cell would be able to activate three different flags, the blue, the green, and the red, then we know that we have succeeded to generate the fetal stem cells. And this is, in order to do that and to find this combination of genes, we had to screen the literature and to do a lot of bioinformatic work in order to find the relevant combination of factors. And what you can see here, it's actually few genes, but if you think about the combinations, it's millions of combinations. We introduced this into the skin cells, and for our delight, and this is maybe the most important result that we got, we could see here that we could activate all the four flags, meaning that we have succeeded to convert the skin cells into the three different stem cell types. And of course, we can isolate these cells now from the culture. And here you can see beautifully how the fetal stem cell look like, how the placental stem cell look like, and how the Yolk stem cell look like. Next, okay, we have cells that look like the real thing, but we really wanted to know whether these cells are functional. For that, we took the fetal stem cell, for example, we stained them by red color and injected them into a developing embryo. If our cells are okay, they function, the embryo should be red. And this is exactly what you can see here. On the upper panel, you can see the embryo and the placenta. This is, of course, mouse. This is not human. And you can see that the embryo is red, the placenta is negative. We did exactly the same experiment for the placental stem cell, but this time we stained the placental stem cell with green. And in the bottom, you can see that the embryo is negative and the placenta is positive with green. This indicates that not only they look like the real thing, they also behave properly and they are fully functional. And this is great. However, we really wanted to know what happened inside the cells, because in order to get to transform a cell, you need that its genome will be modified accordingly. And for that, we joined forces with Dr. Oren Ramlab, who is expert in genomics.
Okay, so we know that the cells look like the real thing and they function like the real thing. But the question is if underlie, they behave on the molecular level the same way. So this is a cell. And inside the cell, in the molecular level, we have the DNA. We know that all the cells in our body contain more or less the same DNA sequence. But we have cells which function like a brain, a liver, a heart, fetal cells, placental cells. So what makes these cells different from each other? So in fact, what makes those cells different is a regulatory layer that open and close the DNA in specific region. Each cell type function specifically because of this regulatory layer, and here you can see a protein that binds the DNA and regulates its property, whether it will be open and or close. So what you see here is a DNA, and you see two genes, gene A and gene B, but apparently in order for gene A and gene B to be operative, we have other regions in the genome called enhancers. These enhancers function like a remote switch. When they rewired correctly, they can turn genes on and off. So this is a regulatory layer that in order to be sure that the cells behave correctly, we need to measure. And eventually, if they regulate correctly, they will make gene A function to produce mRNA out of it, or it will be closed, like gene B. So how will we do it? We take the cells, as you can see here in the upper part of the slide, and we have the targets. The targets are those enhancers that we would like to measure. We break the cells apart and break the DNA into small pieces. Then we use specific antibodies that recognize these enhancer targets. And after fish these targets, we can remove everything but the DNA and sequence the DNA to a reference genome. In that example, the mouse genome. But this process produces huge amount of data. And in order to be able to start processing this data, we need a computational biologist. Professor Tommy Kaplan did that for us.
Thank you, Oren. Okay, so basically Oren produced millions and millions of DNA sequences. Each one of them look like the one you see at the top. They are similar to the ones that Moran described earlier. And then here he had 1 billion different DNA fragments that were sequenced, and we wanted to analyze them in order to understand 3 things. We wanted to know what regions in the genome are open in each of the different cell types. We wanted to know what regulatory regions or what enhancers or switches are on in any of the different cell types. And we wanted to see what is the outcome of that, what genes are active and which are not. So the way we do that is we use computer science, and we basically analyze those DNA sequences, and we compare them to the sequence of the genome. And this allows us to map or to align those sequences back to the genome so we see where they came from. And what you can see here on the left is we can see that open regions tend to accumulate many of those sequence reads. Okay? So the regions that Oren got in his experiment, we mapped them back to the genome, and we can see that some region got more sequences than others. So this indicates that those regions were open. Similarly, we can use another experiment that shows what switches are on and where they are located in the genome. So in a sense, this is the most important things in genetics. We understand coding regions. We do not understand how genes are regulated. And the last thing, we could map the RNA fragments, and we can see what genes are active and which genes are not in each of the different cell types. So let me just try to bring you together back. So again, what we did, we had the skin cells, and then we have the embryo-derived placental stem cells and the embryo-derived fetal stem cells. So those are cells that we know that make the embryo. And now we wanted to use genomics as some microscope in order that allows us to look what's going on inside the cell. And then we did these experiments, and we basically map all the different cells into some abstract mathematical the graph represents an abstract mathematical space where you can see that all the different experiments with the skin cells, they all cluster together. So this suggests that the internal of the cell is similar. We did the same for the embryo-derived placental stem cells, and you can see that they look similar to each other, but they look different from the skin cells, and the same go for the fetal stem cells. Now the big question is, what happens to Yossi's cells? Yossi took skin cells and he wanted to derive them and to transform them into placental stem cells and fetal stem cells. And the question is whether he succeeded not only in the outside look, the way the cells look and the way the cells behave, but also whether the inside of the cells transformed properly. And this is exactly what we saw. We saw that Yossi's skin cells transformed from the lower left part into the fetal stem cells. So they basically, the inside of the cells look exactly the same and behave exactly the same like the embryo-derived stem cells. And the same was true for the placental stem cells, okay? So what are those cells good for? So one thing that Yossi could do with his innovative approach, he can scratch your skin on your way out, and he can clone you, just like Barbra Streisand. And he's not going to do that, right, Yossi?
I'm not promising anything.
He's not promising anything, so be aware. Another thing we could do is we can generate organs, artificial organs out of it. That would be probably done in the far future. In the near future, we want to use this approach to understand how life begins. So we want to understand embryonic diseases, we want to understand placental insufficiencies, and we hope that this knowledge could be transformed to help women that suffer from recurrent miscarriage, for example. Thank you.